This is our hearing. Assalamualaikum, ladies and gentlemen, my dear colleagues, participants. This is the last session of the training setting seminar, and of this session, we have 5 speakers, and the first speaker is Mr. Amir Khan. He is working as an assistant professor at the Pandey Center of Molecular Medicine and Drug Research at the University of Karachi. So, he talked about genomic sequencing and analysis of genomic diversity in the locality transmitted source of the Pakistan. His research interest in human and microbial genomics, their practice publications, he always provides more PhD students, minor cases, own national and international grants, and he is leading the project of sync forensic DNA and experimentally laboratory will provide service to the research system of all proteins in cell. Apart from that, he is a very good human, he is a good gentleman, soft spoken, and always cooperative. Okay, please. And he always, when he interacts with the administrator, that day, today, his face, figure, gene, his gene is constant, as well as his face, figure, and weight is also constant. So with these 3 words, it's a very good friend. With these 3 words, please proceed, Dr. Ishtar.
Thank you very much, Dr. Saab, for very kind and informal introduction. I am going to talk about a very unpopular natural product, which is SARS-CoV-2. As well as the history of SARS-CoV-2 is concerned, based on the molecular clock dating analysis, which is based on the very complex bioinformatics analysis based on the existing species of the viruses. These viruses have been existed since 10,000 years ago, and SARS-CoV-2, as contrary to the normal concept, it is not very unfamiliar to the humans. Before the SARS-CoV-2, around 200 species of coronaviruses were known to humans. And as well as the pathogenicity of SARS is concerned, which causes severe acute respiratory syndrome. In 2002, in China, there was an outbreak of SARS, which is also called as SARS-CoV-1, and the mortality rate of that was up to 15%. Then in 2012, in Jeddah, Saudi Arabia, another SARS virus outbreak was observed, and it is known as Middle East respiratory syndrome, and mortality rate of this was very high. It was around 35%. So, setting aside the conspiracy theories about the origin of SARS-CoV-2, we should accept some basic scientific facts, and that is that lots of gain of research, gain of function studies was going on SARS-CoV-2, and the original SARS was artificially modified in the laboratory, and it was made more infectious. The project was funded by NIH USA, and the collaborating laboratories was Wuhan Institute of Virology, Chinese Academy of Sciences, Wuhan, and University of North Carolina at Chapel Hill, USA. So the outcome was, this study was very interesting. What they did, they took the original SARS-CoV virus and they took the spike protein of SHCO14 virus, which is also a type of coronavirus. They pasted the SARS-CoV spike protein from SHCO14 in the SARS-CoV, and it became very infective. The properties of this new chimeric virus was almost like the SARS-CoV-2. So in vitro experiments demonstrated that it can be expressed in the mouse and in human airway cells, and all the existing immunotherapies and other antibody-based therapies, monoclonal antibodies, was ineffective against this engineered virus. And the work suggested that there is a potential risk, and this study was done in 2015. So alarm bells were already ringing. In 2017, there was another study to understand the SARS-like coronaviruses, and it also concluded that there is a high risk that a new, more infectious SARS-CoV may emerge. We are very familiar with these facts and figures that up to now there are around 426 million cases around the world with 4.9 million deaths. And in Pakistan, there are 1.4 million recorded cases and around 30,000 deaths. The actual pandemic death toll, which is based on the global excess deaths, is estimated to 4 times higher than the reported deaths due to the SARS-CoV-2. So we can say that around 20 million people so far has died due to the SARS-CoV-2. It is a positive sense RNA virus, and there are two main mode of pathogenicity as well as the virulence factors are concerned. There are 3 proteins which are encoded by the SARS-CoV-2, ORF3b and SARS-CoV papain-like protease. These are basically involved in the suppression of the innate immune system. The innate immune system, which is based on the interferon proteins, it is suppressed. And then ORF3, ORF7a, and N protein of SARS-CoV-2, they are the activator of NF-kappa B, and NF-kappa B is involved in the expression of cytokines, and it leads to the cytokine storms, which eventually leads to the different organ damage, as well as it suppresses the immune cells. As well as the outcome of the infection is concerned, there is a wide range from asymptomatic to lethal. The invader is a versatile killer. It can impact your brain, eyes, nose, lungs, and whatnot. Normally we say that the SARS-CoV-2 infection comes and goes, and most of the people remains non-symptomatics. But there are lots of studies currently going on. One is the NIH initiative, which is called as long COVID. And there are lots of meta-analysis based on the different studies which are conducted in different parts of the world. And the outcome of study is that most of the people probably sooner or later, they will experience the long-term effects of COVID-19. Persistent fatigue, for instance, in 58% cases, and weight loss in 12% cases, hair loss in few people, anxiety in 13%, and so on. So there will be a long-term effect in people. The other puzzle which is being observed is that how the super immunity is coming. There are some people which are infected by the SARS-CoV-2, and then when they are vaccinated, so they become super immune, but it is not widespread. There are people which are, posing a greater immunity and resistance, and lots of people are not. Heart disease risk, it soars after COVID even with the mild cases. So in a recent study which involves around 2 million medical records and lots of meta-analysis and comparisons, it revealed that even in mild cases, the existing baseline risk of the cardiovascular disease has been soared up to 70%. So it doesn't mean that we are more, 70% people will, for instance, die due to the cardiovascular diseases triggered by the COVID-19. It is not the case. Every person has a baseline, risk of cardiovascular disease. Some people may have 0.1% risk to have a cardiovascular disease up to the age of 60, for instance, but that will be substantially increased in even mild cases. Viruses, as we all know, that they rapidly evolve due to the pandemic and due to the globalization. You know that this virus SARS-CoV-2, it had two main advantages. Number one, the mode of transmission. It was being transmitted due to the air droplets. So the mode of transmission was very easy. And second is the globalization. For instance, 100 or 150 years before, it could have been rarely gone to that extent. But due to the globalization, it moved rapidly and swept the whole world. Mutations in SARS-CoV-2, it was expected. Initially, people thought that it will probably slowly mutate and the mutations will not that impactful, but it proved wrong. At the start of the pandemic, dominant mutation D614 combined with other mutation started making this more infectious as compared to the Wuhan strain. And then there are some other deletions and insertions were observed, and which made it more resistant to the monoclonal antibodies. So we have seen that there are lots of reinfections. So vaccines, they give a protection against the severe disease, but reinfections almost cannot be avoided. So here is a chart which shows that how different vaccines have been impacted due to the new emerging strains. So the most effective one of all vaccines remain the Pfizer vaccine. Currently, people are scrambling to develop and devise vaccines which are targeted to Omicron, but up to now, the findings show that the vaccines which are developed specifically based on the Omicron, they are not providing greater protection against the Omicron virus as compared to the normal vaccine. So it is being proved as less used. Genomic surveillance in case of any pandemic is very important because it helps to devise the strategies to control, prevention, tracing, social distancing, and so on. So all of these strategies are devised based on the genomic surveillance as the data. Pakistan is a success story, and NCOC, the National Coordination and Command Center at Islamabad, that was the principal seat to combat and devise the strategies to control the SARS-CoV-2. So the base of the success was that the NCOC very quickly recognized that the spread of this virus is unavoidable. It is non-stoppable. So whatever effort you provide, you will be unable to stop it. So they adopted to provide a kind of resistance to the SARS-CoV-2, to try as much as possible to slow down the rate of infection so that the healthcare system is not overwhelmed. In some countries, as you know, that strategy was not adopted, and eventually the healthcare system was overwhelmed. And due to this, lots of people who otherwise could be saved were died. Initially, SARS-CoV-2 split it in two lineages, lineage A and lineage B, and lineage B was characterized by a mutation C281440, and this defines basically the B strain. So A strain is basically the initial Wuhan strain, and the B is mutated one. Now there are hundreds and hundreds of different lineages. As you know that the Omicron is lineage number 5 to 9. It means that there were 528 lineages, different mutation carriers before the Omicron. Genomic evolution of SARS-CoV-2, you can see that there are hundreds of strains. As I told you that the Omicron is strain number 5 to 9. There are hundreds, but only few of them were very impactful. UK strain like which is designated as alpha, beta, gamma, lambda, kappa. The delta variant, and these are the mutations you can see over here. So that as well as the mortality rate is concerned, it is now thought that the delta one was more lethal as compared to the other strains, and Omicron is benign as compared to lambda, but it is much more infectious. You know, when the B lineage, which was actually evolving in different part of the world and especially in the Asian Pacific, so I just put few slides to show that how the scientists were able to accurately predict that what we are expecting in the future. At that time, there was no UK variant, South African, Brazilian variant, or Delta variant, but you know, in January 5, 2021, before the emergence of these variants, it was clearly thought that viral mutations will be the cause of the future very, very bad waves. At International Center for Chemical and Biological Sciences, right at the start, when the pandemic arrived, iccbs was the first which indigenously sequenced whole SARS-CoV-2. And we used a general strategy because at that time, capture probes and amplification probes were not available. So what we did, we used the whole transcriptome of the patient, and we just eliminated the ribosomal RNA, and rest we sequenced by using next generation DNA sequencing, and we were able to completely sequence the strain with around 150x coverage. And at that time, there was no mutation in the spike protein. We sequenced around 200 whole genomes in a spatio-temporal way based on our limited budget. We devised a strategy that we picked very selected samples from different hotspot regions of Pakistan and other areas. Most of the samples were from Karachi, but we also collected samples from Lahore, Quetta, Mardan, Peshawar, and Islamabad. So I will skip methodology, and you can see that lots of mutations other than the very impactful mutations like L, R, and other mutations. So we found in our first batch during the first wave, we identified around 122 mutations. Lots of them were singletons, but 28 mutations were recurrent mutations. We identified D614G in the spike protein in 33 samples, and this mutation makes the virus more infectious as compared to the normal Wuhan strain. You can see here that most of our mutations in our first batch was singletons. Singletons means that virus is mutating quite rapidly, and then most of these mutations are not further selected. So here you can see comparison between the samples in May, June, July, and August, and you can see that mutation rate was gradually increasing with the passage of time. And on the other hand, you can see that recurrent mutations were dropping. Drop of singleton mutations were dropping. It means that more and more mutations were spreading in the population. So here you can see important point which I would like to show over here is that before the introduction of UK variant, South African variant, and Delta variant, in our local environmental setting, this was the B136 strain which was basically dominating. In our during the first wave, we identified that B136 strain was representing around the 12.5% samples. In second wave, it was representing around 25% samples. But in the third wave, it was representing around 50% of the samples. It means that in the absence of UK variant and other more infectious variants, probably the B136 strain could have been the dominating strain in our part of the world. Likewise, during the third, fourth, and fifth wave, we continued our efforts to monitor the mutations. During the fourth wave, we sequenced around 90 SARS-CoV-2 samples, and we identified different mutations, especially the signature mutations L452 and T478K for the Delta variant. And interestingly, even at the peak of the Delta variant, we still had B136 strain in our population. Then, as we all know, that Omicron emerged, and there are lots of things. I don't have time to discuss more about the origin of Omicron, but we sequenced around 20 Omicron whole genomes, and they represented all the signature mutations. We identified all the signature mutations which are the hallmark of Omicron, but we also identified around 300 mutations, which some of them are present in the other strains, some are not. So there is a potential that Omicron-like variants will emerge in future, and especially in those parts of the world where the genomic surveillance is not very strong. And another important aspect is that just recall the first study which was conducted in 2015, and you can see that normal SARS-CoV strain and the SCH014 strain, spike protein was picked from that, and it became very, very infectious. So just look into the possibility that MERS virus, which is the Middle Eastern SARS-CoV basically, it had 35% mortality rate. So just imagine a marriage between MERS and SARS-CoV-2's Omicron variant, and see the impact on the global health. If we have in future a variant which have 35% mortality rate and infectious rate as of the Omicron, that you can see the impact. So as a few days back, Bill Gates also said that we should be very well prepared about the next pandemic. And I picked this slide and this very interesting sentence that Omicron really shows us the need for humility in thinking about our ability to understand the process that are shaping the evolution of viruses like SARS-CoV-2. Probably scientists, lots of scientists, very expert in the field, say that probably we will never know that what was the origin of Omicron and probably of the SARS-CoV-2. Thank you very much. Few seconds for acknowledgement. It was a great miss, basically. I'm very thankful to my colleagues, Dr. Shakil, Dr. Irfan, and my PhD student Zeb. They actually did all this work, and it was really a very hard work. And I'm very thankful to our collaborators from different parts of the country, which provided samples. Thank you very much.
Thank you so only one question. All these available? Okay, one question.
Hi, thank you very much for a wonderful talk. I have a question about Omicron. There is the probably wishful thinking that Omicron is less severe than all variants. But is this more wishful thinking, or is this because we have better protection, more vaccination, and so on? What's your view on milder variants in the future?
Yes, it's a very interesting and important question. My point is that yes, you are right that we have good protection due to widespread vaccinations, but in the countries where the vaccination is not that good, in those nations, even the mortality rate is not that high. It means that Omicron is probably not that pathogenic as compared to the Delta variant. As well as the future variants are concerned, yes, threat is there, and the pandemic is not over. And we should not benchmark the Omicron. Omicron will surely mutate, and it can have very, more devastating impacts, especially keeping in view the recombination potential of this SARS-CoV-2. Different strains, they can, recombine, and due to recombinations, severe variants can emerge in the future. So right now, yeah, Delta variant is present, and there is a risk that due to recombination of Delta and Omicron, a more infectious and more pathogenic variant can emerge.
Thank you, sir.
Thank you.
Thank you very much. So, second speaker is Non-Steroidal. He is having assistant professor at the Pandemic Center in the Applied Medical Research University of identification of non-steroidal aromatic inhibitors and approach to the treatment of breast cancer. And she has 14 research publications, infected 48, and 95 citations. So, her research interest is biochemical activity and potential of inhibition in lens. She also done a postdoc training in the field of structural biology by using animal and computational studies at the University of Hamburg, Germany. These three words, please proceed.
Thank you so much, Dr. Mulis, for your kind introduction. Assalamu alaikum and a very good morning to all of you. The topic of my talk today is identification of non-steroidal aromatase inhibitors and approach towards the treatment of breast cancer. And this talk is basically another dimension of the project, the presentation that has been presented by my mentor, Professor Dr. Muhammad Iqbal Chaudhry, in which he highlighted the identification of steroidal inhibitors of aromatase. And we have done that through biotransformation process, and we got some very promising results. But we cannot deny the fact that non-steroidal aromatase inhibitors are equally important to treat ER-positive breast cancer. So today I'll highlight another important aspect on which I'm working along with Professor Aqiatul Wahab and Dr. Iqbal. Before going into the detail of my results, I would like to give you an update about the breast cancer. And according to World Health Organization, in 2020, breast cancer was the most common type of cancer that was being diagnosed. So the rates are very high, the incidence and mortality rate of breast cancer all over the world. And recently, in January 2022, it has been published by NBCC in a report that in every 13 minutes, a woman dies from breast cancer, which is a very high and very alarming situation. And this is currently the second leading cause of cancer death for women in US and all over the world. And estimated death rates of in 2022 are 43,250 women and 530 men are at risk of their death due to breast cancer. So, why we are more interested in ER-positive breast cancer? Normally, there are different types of breast cancers, including ER-positive, HER2-positive, and triple-negative breast cancers. But the rate of the breast cancer cases that are being diagnosed, among them, ER-positive has the highest rate. If there are 3 cases reported, among them 2 are ER-positive breast cancers. And when I say ER-positive breast cancer, I mean that there are estrogen receptors on the surface of cells that bind to estrogen and transfer a signal for the cell to divide. And what these estrogens are, these are basically different products that are formed by the enzyme aromatase and estrogen synthase, which convert different androgens into estrogens, as you can see here, more better picture. These are the androgens which are being converted by aromatase into estrogens that transfer a signal for cells to divide and reproduce. And this rate limiting step in the intratumoral production of estrogen is basically catalyzed by the enzyme aromatase. So we are more interested to to find out the inhibitors of this enzyme. Because the aromatase inhibitors, they can block the conversion of this androgen into estrogen, and thereby they can help in the treatment of ER-positive breast cancer. In the protein data bank, there is only one PDB ID, that is 4KQ8, which is from the recombinant human aromatase, and rest of all other extra structures belong to the placental microsomal aromatase. So just to show you the active site, this is the androstenedione, and this is the heme, which is the cofactor, and this is your major active site. Aromatase is not the single enzyme. This is basically a complex of enzymes, and both of them are present in placenta lucidum. And this NADPH reductase initiates the reaction by catalyzing the transfer of electrons from NADPH through FAD and FMN finally to the heme of the aromatase, from where they are being transferred to the substrate for the production of estrogen. So these are the currently used clinical drugs. Anastrozole and letrozole are the non-steroidal aromatase inhibitors, and exemestane is the steroidal pharmacophore that is being used. And three of them are being used, but letrozole has the highest affinity or potency for aromatase. The strategy that we have designed is to start from the in silico screening of various compounds that are being submitted in our molecular bank, including various drugs and commercially available natural and synthetic compounds. We have initially started from that, and we will inshallah extend this in silico screening. From that, we identify the hit compounds on the basis of their significant docking score and binding energy, and we also looked into the interactions of these compounds with the catalytically important amino acid residues, as there are various X-ray structures, and we know the catalytically important amino acids. After that, these compounds were proceeded for their in vitro aromatase inhibitory activity. And as you have looked into the talk of Professor Iqbal, that we have worked with UPLC-based method, but in this case, we try to develop a 96 well plate assay, which is more feasible to identify number of compounds in short period of time. So we tried a fluorescence-based assay, and we developed that by using MFC, which is a fluorogenic substrate of aromatase. We identify the IC50 values of these compounds, and then we further proceeded them for STD NMR studies. And STD NMR studies, for the STD NMR studies, we have used the microsomal suspension as a source of aromatase enzyme, and finally performed the cytotoxicity of these compounds against these. Now this is the active site of the placental aromatase, which has a PDB ID of 5JKW, because we were using placental aromatase as a source of enzyme, so we decided to work with this PDB ID. Although the sequences are similar, you can use either of that. And this is your substrate, and behind that you can see this heme binding pocket. So these are the catalytically important amino acid residues. Among them, methionine 374 is the most important. We initially docked the three inhibitors to analyze the mechanism, although we have an idea where they bind. So this is exemestane, anastrozole, and letrozole. Both of them were involved in hydrogen bonding interaction with methionine 374, while letrozole was more interacting through hydrophobic interaction. We next proceeded for the commercially available natural and synthetic compounds, and we selected some of them for the in vitro screening initially. I'm showing you just some of the representative compounds. This is nicotine that was able to form a hydrogen bond with the same amino acid residues with which the aromatase inhibitors are interacting, that is methionine 374. Similarly, this sesamol was also involved in interaction with methionine 374 as well as with phenylalanine 134 by 5-5 stacking interaction. These are some of the synthetic fragments or compounds. This is compound 117, compound 129. I'm not going to repeat all these because the aim was to look into the interactions and with which amino acids they are involved. They should be catalytically important. We next selected these compounds and proceeded them for in vitro aromatase inhibitory activity. As I mentioned before, that this is a fluorescence-based assay and it is 96 UL. For this, we used human placental microsome. There is a whole procedure to form these microsomal suspension. I'm not going into the details. This is a brief view of the assay that we have performed. We basically incubate the enzyme aromatase with MCF, which is a fluorogenic substrate. We add the different concentrations of test compounds. We incubate it for 5 minutes, initiate the reaction with NADPH that will initiate the electron transfer, incubate the plate for 25 minutes to form the product, and finally we stop the reaction by addition of one normal acetonitrile tris base, and the reaction is terminated, which is finally then read at excitation wavelength of 400 and emission wavelength of 540 nanometers. Of course, when we developed this assay, we tried to first identify the IC50 values of the 3 available clinical drugs, which can also validate our fluorescence-based assay. And we found that exemestane has an IC50 value of 0.2 micromolar, while anastrozole and letrozole showed IC50 values in the range of nanomolars, and these were in accordance with the values which are reported in literature. So next we proceeded with the compounds that have been selected from the in silico screening. Unfortunately, we don't have very significant compounds right now, but the screening is in progress. So we hope that we will get some better compounds. But to show some of them, this is compound 76 that showed an IC50 value of 17.58 micromolar. Sorry. These all are natural compounds, but these all are weak inhibitors if I compare them with 3 of the inhibitors which are available. In case of synthetic compound, we got some significant results. For example, compound 158 that showed an IC50 value of 0.1 micromolar, at least closer to exemestane, and compound 152 that showed an IC50 value of 2.58 micromolar, and there were some other that showed IC50 values in the range of 11, 12, 18, and the highest was 14 micromolar. So in the next step, we try to study the interaction of ligand receptor, and as we know that in vitro assay do not reveal the direct observation of ligand binding to the target, but STD-NMR is a powerful method for studying protein-ligand interactions. And in this case, we have not used the pure protein, we have used the microsomal suspension as a source of aromatase binding. And by using STD-NMR, you can analyze that how much saturation has been transferred in terms of percentage and which protons are in close proximity with the receptor protein. So this was the aim for which we ran various proton NMR individually, and then this is the proton NMR of microsomal suspension, this is a lone buffer, and then we proceeded with the standard inhibitor to validate the STD-NMR procedure. This is letrozole, and you can see the proton NMR of letrozole. This is proton NMR of letrozole with microsomes, so we can analyze the peaks which are from microsomes, the resonances. Then this is STD-NMR different spectra of letrozole plus microsomes in buffer, that means with aromatase. And this is the reference STD-NMR that was run in absence of microsomal suspension to validate that these resonances were due to binding with aromatase enzyme. And we found that this triazole ring is basically in closer proximity to the protein, as it has highest percentage of saturation transfer. Similarly, anastrozole was also proceeded for STD-NMR studies, and the reference spectra did not show any peak, highlighting again the fact that the binding was due to aromatase, the presence of aromatase. And in this case, again the triazole ring showed highest saturation transfer, indicating close proximity of this part of the ligand and followed by this panel ring. This is one of the ligands that we have identified, and we run the STD-NMR, and we were able to get the resonances that were interacting. And in this case, this is the major part which is in close proximity to the aromatase. This is another one in which you can see methyl group is in close proximity to the protein and then followed by this part of the ligand. So this was basically in itself a time-taking procedure, because we need to develop the the STD NMR using microsomal suspension as energy loop. There could be many peaks, many resonances. So it took a lot of time to optimize the STD parameters, and once they were optimized and validated by the clinical drugs, it was easier for us to identify the ligand receptor interactions of all other ligands that we have identified, and there are many others. So in conclusion, we have screened more than 1,000 compounds using in silico studies against the crystal structure of human placental aromatase, and then we selected 250 compounds initially on the basis of docking score and negative free energies. We proceeded them for the in vitro human placental aromatase inhibitory activity. Among them, 3 drugs which I haven't shown because they were not that significant, and 52 compounds showed IC50 values in the range of 0.1 to 82 micromolar from significant to weak. And these 20 compounds were then proceeded for cell-based STD NMR studies, which further showed their interaction with aromatase. And finally, we proceeded them for cytotoxicity studies against BJ cells, the human fibroblast cells, and they all were found to be non-cytotoxic. So we are going to further extend this screening because initially we were in the stages of developing in fluorescence-based assays, and that took a lot of time. I would like to acknowledge my mentor, Professor Dr. Muhammad Iqbal Chaudhry, and my teacher, Professor Dr. Atiyatul Wahab, who helped me a lot in whole of the project, and Rabia Nees and Nimra, the students who have worked in this project. Thank you very much for your attention.
Thank you. Thank you. Thank you very much for your presentation. And due to time constraint, the question is coming. So, only one question.
Thank you. Very nice talk through your work there. I always like to see a pipeline, like you showed in the early slides. But I wondered why you chose as the third step to do NMR and not cytotoxicity tests. Because I would logically, personally, think cytotoxicity should convert.
Yeah, I haven't shown that initially, but of course we first proceeded after performing the in vitro assay. We normally proceed our compounds for the cytotoxicity data, and then we finally take them to the STD NMR studies because all of them were non-cytotoxic, so we proceeded them for STD NMR studies.
Thank you. Thank you. Quick. So, final question. Sumaya Jacob. Sorry, Sumaya Jacob. Sumaya Jacob. She's assistant professor at Dr. Panjali Center of Molecular Medicine and Drug Research. She talked on identification of the new lead molecule anticancer drug target T2878 using artificial and molecular protein studies. Her research interest is molecular and structural biology, biochemistry. She has 15 years of contribution to the high impact journal, international journal, Our research includes contribution of compounds which possess the potential of inhibiting enzymes or expressed in various animal conditions. So after her PhD, she got interested in molecular training on identification of enzyme mechanism in series, myelin, and she was worked at the Institute of Pharmacology and Structural Biology, France. At this moment, she was working on structural biology using more restrictively information on isolation of new things and less different biologically important proteins, biophysical and biochemical methods. To these keywords, Dr. Sumaya Jacob. Thank you.
and the TH complex protein PH. And I will share this presentation with my teachers, Professor Dr. Ajay Wahab and my mentor, Professor Dr. Who will basically train me in the field of molecular biology and biomolecular NMI factors. So let's start why I want to work on this target. Basically, I wanted to work on this complex because this complex has a role in the production, in the process of transcription, which has to be found to be upregulated in cancer at a very high level. Cancer cells are known to be addicted to the high levels of the transcription and DNA repair. This is the basic reason I want to work on this transcription factor. This transcription factor complex basically comprises of 10 subunits, after which 3 subunits form the cyclin and protein kinase complex, which has a role in the production in the cell cycle control, while the core contains the 7 subunits, which basically has a role in the nucleotide activity. Together, these 2 complexes have a role in the transcription of the genes. Compounds that affect the activity of various subunits of this complex have been found and developed as a strong candidate to combat cancer. However, there are some molecules still in the clinical trials. This slide basically shows potential druggable targets of the TS-based complexes. We can basically disrupt the protein-protein interaction of this complex by either inhibiting the activity of this PDK7 kinase enzyme, either the SPD, XBP, or by disrupting the interaction between the P34 and P44 and by interaction of P8 and P52 molecular protein interaction. I choose to work on this because this subunit has a very important role in maintaining the structural integrity and concentration of this complex in the cell. This subunit exists as a dimer. It is required for stabilizing the cellular concentration of the complex. It is found to be frequently upregulated in severe cancer patients, and it basically promotes cell proliferation and inhibits the apoptosis. Our idea is that if we can find out some compound which can destabilize protein, those compounds would be expected to destabilize the complex, and therefore we can have some role in combating the cancer. So to start the process, we should have the protein in hand. For that, we actually follow the recombinant technology. We express that protein in the prokaryotic system, for which the system is already established. We clone the gene in the PET20A vector, and then we transform this plasmid in the bacterial strain BF21, and then we check the presence of the transformant in the colonies by colony PCR and perform the gene sequencing to verify that the gene which is inserted in the plasmid is correct or not. Then we check the expression analysis at a low scale and then at a large scale. And after we found that the protein is expressing at enough amount, we took that to the affinity chromatography and size diffusion chromatography, and we performed the just to confirm that it is coming at the right position. We check the folding of the protein by proton and mass. Then we took that protein to the biophysical method, which basically I have used STD NMR and thermal shift assay, as various speakers talked about the STD NMR. That is one of the most important techniques to study the protein ligand interaction, where you basically saturate your protein at certain position, and if your level is present in close proximity to your protein, it will receive that saturation and you will get some resonance in your basic spectra. In photoshop assay, basically we actually see that either upon the addition of your protein, either addition of your compound, your protein remains thermally stable or not. So this is the overview of the study which I am going to explain you. We have done the screening of compounds in the form of mixture, where each mixture contains around 4 to 5 compounds in it via STD NMR technique. From there we have found that 13 compounds they showed a potential. They found to be potential in the binder, and these binders were then validated individually and we found that only if were able to retain their ability to interact with the protein. These compounds were then taken to the SCD-NMR competition experiment to show the binding site of the ligand on the protein. Then we performed some molecular blocking to get some structural insight into the protein with interaction. Compound identified from these studies were then taken to the synthesis assay to study that whether they are destabilizing or stabilizing the protein. Finally, some titration studies were done to determine the affinity between the ligand and the protein complex, and at last, the cytotoxicity. So I'm going to start my result discussion by describing the mixture one. This is the proton NMR analysis of the mixture one, which basically contains the four compounds. This is the proton NMR of the mixture, and these are basically the proton of different compounds alone. When we did the SCD-NMR SCD of this mixture, we do not find any interaction in the. This is basically the proton NMR of the mixture. This is the different spectrum of the mixture alone and in the presence of the protein, and they found that there is no compound interacting with your protein. So this means that mixture one is not able, any compound from the mixture one is not binding to the protein. Then the same result for mixture 2 to 5. We actually screened 100 compounds that was grouped in 20 mixtures. Okay, so the similar result for the mixture 2 to 5, which contained 4 to 5 compounds, but no interaction of these with the protein. Then mixture 6. In mixture 6, we got some resonances that were found to be present when your sample contained the mixture and your protein. When we align these resonances with the proton NMR of the compound alone, we found that these resonances are basically coming from these two compounds. We next perform the SCD-NMR analysis of those compounds individually, and we perform the epitope mapping of these compounds. This is the compound one which did not show any interaction when it is done alone in the absence of the protein, but when it is done in the presence of the protein, we found that these protons are actually interacting with the protein, so these interactions are basically originating from your protein. As Dr. Humera told you, that we assign the proton which is giving the highest SCD intensity to be 100%, and we normalize other proton according to that. So we found that the protons which are present in 9, 11, and 13 region, they are basically present in close proximity to the protein, while the other protons, they are also present near to the proximity of protein. Then we did the STD competition experiment of this compound with the reference compound. The STD competition experiment are very nice experiment, very strong approach in which we can basically probe the binding side of that ligand to the protein if you already know any other ligand. It's just like enzyme kinetics. So what we do in this experiment is that we done the STD-NMR experiment on the reference compound and on the ligand compound. If the resonance of those compounds disturb the presence of other compounds, it means that both are competing for the same site. If the resonances of those two compounds, I mean the reference compound and the unknown ligand compound, are same in the absence of the other or in the presence of the other, it means that they are not competing with each other. So we found that in our case, the resonances of these two ligands are not disturbing the presence of others. It means that the reference compound which is already present, I mean by reference compound, I mean the standard compound which I will see in the protein, is not competing for the ligand that we have identified. So similarly, the story for the mixture 2, where we found some more compounds that are interacting with the protein. The epitope mapping of the compound 4, which we identified from the mixture 40, and we found that this proton may be, as it is showing 100% saturation, it is present in close proximity, followed by this, and this chain, the aliphatic chain, is present a bit away from the protein. So this is the STD competitive experiment of the compound 4 with the reference compound. Again, we did not find that the compound and the reactant compound were binding on the different. These are the 8 molecules which we have identified through this approach. These compounds were then taken to the thermal shift assay, where basically we found that either the presence of compound is inhibiting, is thermally destabilizing the protein or not. This is the this is the PM of the protein when it is not, when it is present alone, and when it is present in the presence of this compound. And we found that most of the compounds, they are causing the destabilization of the protein to some extent. And based on the highest destabilization caused by this compound, we took this compound further for the acidic calculation studies, and here we found that this compound has an affinity, has a KD of 1 millimolar, which we determined using this formula, and it was found to be a bigger binder of this protein. Now I'm going to tell you a bit about the structural characteristics of the GF protein. This is basically a dimer protein, homodimer protein, is comprised of the alpha 2 alpha C and beta sheet, and you also have a hydrophobic pole present in the vicinity of this protein, which has a role in the stabilization of this protein. During the molecular docking studies, we found that most of these compounds are actually interacting with the protein at the hydrophobic pole, indicating that they are basically interacting with the important amino acids which are present either at the hydrophobic pole or the dimer side of this enzyme. So this is basically the dot code of these compounds, 1 to 8 on the dimer. These compounds are supposed to be found near the hydrophobic pole of the dimer. The molecular testing studies were performed by advanced of the mineral transfer using the microscope of Dinger. So this work has been published last year in Bioorganic Chemistry. And in conclusion, I can say that the structure of protein infection in CF2 complex by targeting the pH subunit is a promising way to reduce the functional role of the CF2 complex in the cancer. And here we have identified 8 non-cytotoxic molecules which account for the destabilization of the protein, and these compounds with a new step will therefore hold an interesting perspective in designing new molecules which can be possible in cancer. With this, I would like to acknowledge my teachers, Professor Rahman, my mentor, Dr. Chaudhary, Professor Abdul Wahab and her group, Dr. Humaira Zafar, my students, and all technical and non-technical staff of the ICCBS. Thank you.
Thank you very much. Thank you very much for wishing a good time. So the house is open now for 1 or 2 questions. If there is no question,
but I've got one question. But this stabilizing the protein, which is usually this is using excess. That is only possible that the compound opposed to the reaction is actually binds to the genesis of the diamond, but the binding formation. Well, that bind is quite binding, so
yeah, even if it is binding at the dimer site, this would be advantageous for us because this protein basically exists as a homodimer and as a heterodimer. Upon getting the signal that it should be involved in the translation, it dissociates the homodimer, and it gets associated with one of the subunits of translation factor towards that is ICCBs. And there is the
as a follow-up question, if it's a dimer or monomer solution,
yes, it is found to be, I haven't performed the dimerization site in using the advanced one that we have performed before, it is basically a bit in the diagram. Because the relaxation, the time it, correlation time it takes to move is about 15 nanosecond, which corresponds to the dimer correlation.
Thank you very much. It was a very good presentation. Just want to know, you show that reaction minus the gate compound. Which compound you use in the precursor?
I haven't synthesized these compounds. I have taken these compounds. I performed the random screening of the compounds on the various levels of the.
So you use the library?
Yeah, library is the one you are already finding.
Thank you so much. Thank you very much. So from the organizer, we have a book. Fourth speaker of the session is Dr. Maria Muhammad. The assistant professor of the University of Connecticut. She taught on mainly biodegradable transistor and natural surfactant and alternative scientific approach. So her research is chemistry, nanochemistry, environmental and studies. So she did PhD in December 2020 and she got the best researcher award in 2021. She published 9 papers with first paper in 2042. She was supervised 118 and she PhD student. She is a personal audit, a member of the committee, and she also established different labs for BS and MS level and 4. So, Dr. Maria talks. Please proceed. Thank you.
Bismillahirrahmanirrahim. Good afternoon and a very good day to all of you. This is Dr. Maria, as he gave a very nice introduction about myself. So, from past 3 days, we have learned a lot about natural products. Natural products for the benefits of against against diabetes. Natural products for the use of different against carcinogenic effects, against antiviral, antibacterial, antimicrobial, and so on and so forth. You just name it, and natural product will do something good for you. So one thing which were missing in all these 3 days and all these even today were the natural product in case of surface chemistry, in case of surfactant sciences, in case of surface chemistry. So I wanted to bridge this gap because none of us discussed, or maybe I missed, maybe somebody discussed it, that natural product in the case for the purpose of surfactant. First of all, I would like to briefly introduce about the surfactant, that this is not anything new. This surface active agent term is very old and it is coined in 1915. But why surfactants are so much important is because if I just simply say you that we start our day with the surfactant and we end our day with the surfactant. This is very good phrase to know, but in reality, it is like when you brush your teeth, when you wash your face, when you wash your hairs, you are consuming many different type of surfactants, and not only for the dermal purposes, not only for the face wash purposes, but you also consume some surfactants in your milk, in your juices, and many different other food products. And at the end of the day, and continuously we are consuming different natural different surfactants in whole day. And at the end of the day, when you go, before going to bed, you do some cleansing and some washing and stuff like that. Again, you are using different surfactants. So surfactants should be very much economical, first of all, And secondly, they should be very much human friendly, eco friendly, environmental friendly, equated life friendly, means they should be good for the consumption. So why surfactants are so much important? Because they help you to interact between two different phases. They reduces the interfacial tension, because of which you can easily interact with different substances from one phase to another phase. That is why surfactants are very important. Surfactants are very simple before its CMC point, but at the time of micellization, at the point of micellization, when all the single monomers become combined together and they form something like this is called as micelle. And at this point where micelles are formed is called as critical micellar concentration. We have two different critical micellar concentrations. One is CMC1 first point and CMC second point. Second point is a connection between two regimes, and it can be of different shapes. So surfactants are basically surface active agents. They reduce the surface tension and also they improve the solubilization. And because of this property of surfactants, that they improve and they initiate the solubilization, we can use it in many different industries, many different places. The uses of surfactants are so wide; they are used in many different industries, like leather industries, like petroleum industries, like cosmetic industries, fabric industries, textiles, cosmetics, perfume. You just name it and surfactant will be there. There are many different types of surfactant. As I already told you, there are so many uses of surfactants. So definitely there are many different types of surfactants as well. Surfactants can be synthetic, bio or natural. Synthetic surfactants are of many different types. They are ionic, normal, Bola, Gemini, polymeric. And among all these surfactants, Gemini surfactant is most good for the purposes, like they have a very remarkable aspects. But the problem is the Gemini surfactant is the most hazardous among all these kind of surfactants. So then we have another thing, like we have biosurfactants and natural surfactants. Natural surfactants in which we have plants, we have animals and microorganism based surfactants are there. If we talk about some basic harmful effects of the surfactant, it can easily cause you skin irritation. Okay, first of all, I would like to discuss something. Have you ever listened about SLS free shampoos when you go to some market, when you go to some places, and they used to promote you to buy their products? So they say that this is SLS free, this is SLS free. So you feel like, I don't know what is this. So this is because SLS is something which is called as sodium lauryl sulfate or sodium rhodocyl sulfate, which is a synthetic surfactant. It can easily cause you skin irritation. It can cause you dandruff in your hair. It can easily irritate you very easily. And I guess all of you have experienced this, that early appearance of a gray hair is because of high usage of shampoos. And why? Because all these shampoos contain SLS. Most of these shampoos contain SLS. And now we are finding some alternatives. So they can cause skin irritation, damage to enzyme activity, damage to liver, carcinogenic in nature. So we have very many different harmful aspects of surfactants. Surfactants are not only bad for us, but they are also very harmful to the aquatic environment, for the aquatic life, for the marine life. Why so? Because as I already told you, the surfactant increases the solubilization of any substance. If it increases the solubilization, it do increase the solubilization of many pollutants. They do increase their availability in the environment. They do increase availability for the aquatic life. So when you dump your wastages into the aquatic system, they definitely increases their solubilization for the marine life, for the animals. So it is also very harmful for the aquatic life, for the environment, and for us. The applications of surfactant, as I briefly discussed before, is in the field of antibiotics and pharmaceutics. There are so many applications of surfactants in the field of antibiotics, in the field of pharmaceutics. Why? Because we want to increase the solubilization into the body. So we do want to increase the solubilization of medicine into the body. That is why you encountered many, as I already told you, that we consume surfactants because of this. We do consume surfactants. Because of this, we do consume surfactants. So So it is because they increase the solubilization, so we do consume it. Okay. As I said many things about surfactants, their benefits, their drawbacks, their harmful effects. So we have some good news. I do have some good news for you that we do have biosurfactants. Biosurfactants are not harmful for the environment. Biosurfactants are not bad for the human consumption, neither for the aquatic life. There are approximately 100 plus families which belongs to such compounds, such natural products, because this conference is about natural products, I'll use the term natural products. So they do contain natural products which have surfactancy, which do have detergency, which do have cleaning abilities, which do have solubilization properties. All these contained by these 100 plus families of compounds. So we should know about it. Okay, little bit I want to discuss about dyes as well. I'll connect it with the surfactants after 1 or 2 slides. So dyes are, you all are, if I'm not wrong, most of you are from organic chemistry side, so you all, major of you are organic chemists. So dyes are organic compounds, they are aromatic in nature. Again, they have many different applications, like they are used in tanning industries, in textile industries, in fabric industries, in cosmetic industries, in paints. In all these industries, we do use dyes. And again, dyes are also not very good for the humans as well as environment. Oh, so I should skip these all. And why do we use dye surfactant interactions? Because dyes are, as I already told you, that dyes have many different industrial aspects and all, and surfactant do also have. So if we interact both of them, so we can easily target many textile industries, many paint industries, many fabric industries. That is why we do dye surfactant interaction. That's how dye interacted with surfactant, like this. Before CMC point, as I already discussed with the CMC point, it interacts with its palisade, and after the CMC point, it reaches to its core. This is basically the dye surfactant interaction. It is not a proper bond. If there is no ionic or covalent bond, but it is attraction repulsion bond. It is the weak van der Waals because of weak van der Waals, because of hydrogen bonding, because of pi stacking, because of all these attraction bonds, they do interact with each other. We do use this ion pair property for extraction of many different such compounds which are not easily extracted in different phases. This kind of ion pair is very much common in oppositely charged dye surfactant, but very uncommon in case of a similar charged dye surfactant. As I don't have much time, otherwise I would love to discuss it. In this study, I discussed we took some natural product compound like acacia. We took acacia from the local market, and we took its extract for it to know its surfactancy. It is a high molecular weight compound. Its saponins are 1.58 molecular weight. It is acidic in nature, but with one replaceable proton. And look, its CMC is 4.4 exponent -4, which is quite low. I'll discuss it. Look, here we have two different surfactants, as I already discussed. SDS, which is well known, highly used surfactant, which is also called as sodium lauryl sulfate, sodium rutile sulfate, and commonly we do say it as this. And it has another surfactant which is also highly used surfactant. These both surfactants are synthetic in nature. This one is anionic surfactant, this one is cationic surfactant. On the other hand, our natural product, our natural compound is having the properties of both cationic nature as well as anionic nature, as I discussed in the previous slide. You can see that its CMC is 8.3 exponent -3. Its CMC is 1 exponent -3, but its CMC means its point where all the monomers gather together and they show their best ability towards the cleaning, towards the solubilization, is a 4.4 exponent -4, which is far lower than these two surfactants, which are synthetic and widely used. Okay, our single natural compound, single natural product is able to interact with the cationic dyes as well as anionic dye. On the other hand, CTAP can only interact with the anionic dye, and SDS can only interact with the cationic dyes. Only opposite interactions are possible, as I already discussed. You can observe from this table that we have tried many different dyes, and our natural compound is able to interact with both cationic and anionic dyes. This is its interaction spectra. You can observe that its ion pair is formed at 5.5 and 5.4, which is far lower from the ion pair formation which SGS and CTAP can provide you. SGS form ion pair at 8.3 x minus 3, where CTAP form ion pair at 1 x minus 3, which is far higher than this. Means low consumption, better results in case of our natural surfactant. I don't have, I guess, much time to discuss this spectra and this spectra. These are also spectra which shows that activity and behavior of our natural product, which is far better. Okay, our natural compound is biodegradable, self-degradable. We do not provide any medium, any substance, any media to the compound, and it's self-degraded. So you can observe that in spite being having so many good things like good interactive abilities, good point of micellization, ion pair formation, it is also biodegradable. And again, one more thing is that it is a single natural product which can replace both types of synthetic surfactants like cationic surfactant as well as anionic surfactant. So we have win-win situation in this case. Advantages of biosurfactant, as I already discussed: low toxicity, good tolerance, good surface activities, good solubilization, eco-friendly, human-friendly, bio-friendly, biodegradable. So this is some work which I published from the presented work. Thank you for your patience and your time.
Thank you very much for a nice presentation. And very interesting. So how do you finish? What one of the questions?
Yeah, thank you. It's obviously very important to reduce the chemicals in the environment in our lives. Pollens, as you all know, are very abundant, easy to extract and isolate. But they do have some toxicity. I know they affect local business to taste of animals and plants and protect it. I just wonder if you could comment on the potential of concentrating pollens in the environment to temporary, could that have a toxic effect in a local area?
Their toxic effects are very much lower as compared to the toxic effect of a synthetic surfactant. I'm just saying that we can replace synthetic surfactant with the natural surfactant. I'm not saying that this natural surfactant, as it is a natural surfactant, so it is very good, like it cannot harm you anyways, but its harm is much lesser than the harm you are getting from the synthetic surfactants.
Thank you very much. From the other media. Yes, this is the last lecture of this week. The session on media. The last lecture will be delivered by Untitled Chapter. So we can consider the same thing. Shift on XML, polymorphism, RBC membrane, digital membrane. These are classes. Is particularly interested in the implementation of any economic strategy in order to explore the mechanism in the area of disease and changing the metabolism or pathway modulation due to diseases or complex biological system. She did PhD in 2016 and then she joined ICBs and explored various applications of the analytical techniques starting from life sciences and microbiology. We have postdoc in France, we have postdoc training in secularized analysis in the RDNS under the joint scholarship of Campus France and ICBs. She has handled many projects in more than 30 publications in journals, and she has interpreted 122. If you want, please start on the side.
natural products less constant with some biopolymers or biofluids. My presentation is based on the excellent polymorphism along with the hydroxyurea administration, which alters the lipidome of RBC's membrane in beta thalassemia. So many of you know about what is beta thalassemia, but little introduction. This is a disease, genetic disease characterized by reduced or absence of human beta globin genes, and this gene is present on the 11th chromosome, and this genetic mutation results in the few numbers of hemoglobin cells or the RBCs. This results in the anemia or anemic conditions of the patient. And what is hemoglobin? Actually, just a little introduction. This is an oxygen-carrying protein and contains 2 alpha chain and 2 beta chains. Not only this combination is present in the human, but there are other globin chains also present, but in different life stages of the human. Like in the embryonic stage of human, there are 2 zeta and 2 epsilon globin chain. Further, during the early stages of the fetal life, up to the 6 months after the birth, the chain 2 alpha chain and 2 gamma chain are combined to form the fetal hemoglobin for the oxygen carrying. This fetal hemoglobin is much more have much more affinity as compared to the mother hemoglobin, and that's why the fetus absorb oxygen from the mother. After the birth and the 6 months of the birth, this gamma chain is replaced by the beta globin chain of the gene and forms the adult hemoglobin. And this adult hemoglobin is present at the end of the death of the gene. This change or shifting of the chains of the hemoglobin is from at the different stages, and this results in this is due to the affinity which required at the different stages of the life. As the beta thalassemia is a genetic disease, so there are a number of genetic mutations that are reported for the beta thalassemia, and almost 200 mutations are reported for the beta thalassemia. So far, most of them are simple insertions or deletions of single nucleotide. But apart from the genetic mutations, there are some polymorphism also reported for the beta thalassemia. But today I'm discussing polymorphism that is commonly known as excellent polymorphism. But this polymorphism has some ameliorating effect, means it decreases or reduces the severity of the beta thalassemia when it is present in the tissue. So this polymorphism is present at the promoter region of the gamma globin region, and the C is replaced with the D. And okay, so this polymorphism is, I told you that this is good for decrease the severity of the disease. So in addition to the therapeutic approaches that are used for the beta thalassemia patient, this polymorphism should also be considered. So I will link this polymorphism to the therapeutic approaches. So far, the therapeutic approaches that are using in this for this disease. First of all, as the patient is anemic, so you have to give the blood transfusion, regular blood transfusion for the patient, and which of course results in the iron overload and some blood-borne diseases, that is viral infections and other infections. So in order to reduce the iron overload, some iron chelation therapies are need to be provided. So finally, if you don't want to have transfusion or the patient do not accept the transfusion, you have to do the spleen echo. So the most promising approaches for therapy are the hematopoietic stem cell transplantation or the gene therapy. But both of them are so expensive. So many of the patients in our country, like Pakistan, India, they are not good for these two. So in order to replace or in order to find some good therapeutic approach, we go to the HPF induction therapy. As I told you earlier, that there is a presence of fetal hemoglobin in the early stages of human life. So if adult hemoglobin is not working or not performing very well, so we can shift to the early stages of hemoglobin, that is fetal hemoglobin, or use some therapies that can induce fetal hemoglobin to the patient. Fetal hemoglobin induction therapy, there are a number of compounds that can do this induction, but here I discussed the hydroxyurea that is very much commonly used in by the clinician in Pakistan. Almost 70% of the children in Pakistan responded very good to this induction therapy. But still, there are some patients that can not responded very good or even very bad responded to the hydroxy. So there is some clue with the that why some patients are not responded to this induction therapy or hydroxy, while the other of the same population have responded. So here we combine this induction therapy to the genetic mutation, or specifically the XMN polymorphism that gives the amnesic effect of the disease. So combining this therapy with the XMN polymorphism, we can conclude that why the patient have not responded. So our objective is to select the patients that have XMN polymorphism or not have not the XMN polymorphism, and then provided them the hydroxyurea therapy. Some of them have not been administered with the hydroxyurea therapy, and the rest of them provided. We perform the RBC's membrane lipidomics analysis. We extract the lipids from the RBC's membrane, and we know that the major portion of the membrane is lipids, and perform the UHPLC ESINS analysis for whole lipidome. And then link the two good things for the therapy. Okay, this is the workflow. Select the patients, divided into two groups, XMN or non-XMN, and then provided the different doses of hydroxyurea that is required for the patient. Collect their blood, extract the RBC's membrane, and then isolated the lipid from the membrane, and do the lipidomics analysis using the HPLC analysis. So in every omics analysis, there are many co-founders can be performed that can affect your omics results. So we have to identify if there are any confounder present. So the most common confounder for any omics are the ages, different ages, or the different gender of the patients. Because if the gender is different, your omics, specifically the metabolomics or the lipids, there are the final stage products of the biochemical system. They can easily be changed. So we found that there is no grouping or no formation of cluster in the age or gender of the samples. These are based on the lipid profile of all patients. Every dot in this picture is represented a separate sample. Okay, at the next stage, then we obtained that there is no confounder present. So we perform the significance testing of mass spectrum features. These features are obtained with the positive ion mode of the mass spectra, and we perform this significance testing using t-test simply, and the p-values are plotted against the fold change of every feature. These features are not yet identified that what lipids are they, but we find that almost more than 30 features of mass spectrum are highly significant, means they are totally different in XMN as compared to the non-XMN features. Further, these features that are purple in color would be identified using different databases, lipid blast or human metabolome database, and we found that 21 of them are identified with high VAP values, and their fold changes are mentioned. So at this stage, just mark that there are some red lipids. Their VIP values are more than 1. What are these VIP values? I will explain to you in later slides. So these lipids are clustered together according to the XMN and non-XMN polymorphism, and you can see that the intensity of these 4 lipids are totally different in this group and in this group. So there are also some differences present in the other lipids, but they are very clear. Very clear means they are formed in 2 groups, including phenyl valerate, palmolic acid, and choline acid. So we can say that these 4 lipids can be used further for separation of these 2. So further we perform prediction analysis whether our mass spectral features are significant or much are they well enough to perform the grouping of 2 patients. We perform OPNLC analysis, and this is used for the prediction model, and we predict that with 80% specificity and 93% sensitivity with almost 12% misclassification. So this is the features, all features present in the mass spectrum, and you can see that there are few features, few masses that are colored in red. They are the highest VIP values that I have mentioned in this slide. These red ones. So these are the most important ones that can discriminate the 2 groups of the study. This is the validation one. Okay, at the second stage of this study, we add the second factor of our group, our study, that is hydroxyurea administration. So there is a combined study, or this is a statistical test, ANOVA, coupled with a principal component analysis, and this results in almost 15 features that are that can use to classify the 2 groups or the 2 factors for metastatic simulation. These 15, unfortunately, we cannot identify all the 15 lipids, but here you can see that the 2 main lipids of this analysis gives very clear or separated results, like PC041 is very much present, higher amount in XMN patients with the hydroxyurea. So this lipid can be used further for validation studies, as this is very much common. The other one we cannot identify at this stage because of some parameter issue in the mass spectrum. So this is very much higher amount in red one. This red one. These are the samples of non-XMN patients with hydroxyurea treatment, and this is very much low, blue one, with patients XMN and hydroxyurea. So finally, we conclude that lipids can be used to specifically the PC lipids, or that I have mentioned that these 2 factors can be correlated using this PC lipid, And further validation needs to be done, as this is a non-standardized identification. So in future studies, we can use different PC limits as a standard to perform this analysis on a larger scale, and give some chemistry overview of the lipidome for this and mitigating effect of the excellent polymorphism combined with the hydroxy. So I do want to acknowledge this study as Dr. Shamsi, Professor Dr. Shamsi. He made us please continue more with us, but he provides the samples for this study from the hospital. And my mentor, my dear Professor Dr. Surfactant Musharraf, and I am unable to do mass spectroscopy without her. I co-authors for this study. That was gradually. Thank you.
Thank you, Amma. Thank you very much for the nice presentation. So, for the organizers, stand from them. If you have any questions, please put your hand up.
Here I am Dr. Devana. Very interesting, very interesting topic you choose for this study. My question is, in Pakistani population, how many genes you detected responsible for this disease? In India, I studied many genes they identified responsible. So first study strategy should be that identify the responsible genes. So how many
There are a number of genes that are responsible for the beta thalassemia, for the mutation of the beta globin chain, almost more than 200 overall. But in Pakistan, for the clinical identification of the patient, we use almost 20 mutations. If any of them are present, then we can suggest that this is the patient of beta thalassemia.
There are variation, genetic variation of those genes or
no, it depends, because 20 mutations on the whole. If any of them is present, then you can say that this is the patient of the thalassemia.
Thank you, Amma. Thank you very much. For the organizer, ladies and gentlemen, please. So, dear participants, dear colleagues, dear participants, from home and abroad, thank you very much. Hopefully, again and again and again. So, the counseling session will start at 2:30 p.m. sharp at 12 p.m. And all the poster presenters are requested to bring the presentation to the ministry to get the clarity. So now this proceed belongs to the Manufacturing Hall, and our conference will start at 2:30 p.m.